Hello, everybody. Welcome back to the 70 seminar series, organized by Johns Hopkins University and Kennedy Krieger Institute. So this week, as you can see, we have a very exciting topic. As you know, if you have attended previous lectures, we sort of trying to focus the main theme, towards a clinical application or more immediate clinical application, on high fields. But this month we thought, it may be interesting and exciting to talk about what's being developed for the future of high field MRI. So we decided to double the field strength, from 70 to 14 Tesla. And, we're very happy, Dr. Van Kolk agreed to come to talk to us about this topic. So just a very quick, introduction. Dr. Van Kolk is a clinical clinician scientist, working in MRI for many years. She got her training from Utrecht in Dr. Peter Luden's group. And as you may know, she has pioneered lots of work, especially on the topic of intracranial vessel wall imaging at 70. I believe that was the topic last time I invited you for a presentation. And then she has been an expert on ultra high field MRI of neurological diseases. And her current interests include vascular imaging, metabolic MRI of brain tumors, and neural inflammation. As you know, she's also part of the Dynamic Consortium, which is dedicated to build a 14 Tesla MR scanner for whole body in vivo human imaging. And then, which is the topic of today's presentation. So without further ado, I'm really looking forward to your talk. And please tell us, is it really insane or not? Thank you.
Thank you, Jun, for the introduction. Yes, so for everyone who's now starting to get in and starting to listen to this presentation, I'm going to talk about 14 Tesla MRI. And I said, "Are you insane?" I put that in my title because many people actually told us over the last couple of years, "Are you insane going to that field strength? I mean, we're barely getting used to the 70. Let alone 14 Tesla. People will get dizzy. It won't be safe. Why are you doing this in the first place?" So I thought, especially because it's now approximately 1 year since we had, since we introduced the fact that we're going to build this machine, I thought it was time to sort of update you and show everyone what we want to do, what our vision is, what we might be able to do with the 14 Tesla MRI scanner, why we want to do it, and also, is it there yet? It's not, by the way. So D-Day was February 20, 2023. At that day, there was a news item on Dutch researchers that plan to develop the world's strongest MRI scanner. It's not really the strongest MRI scanner, of course, because there are several other MRI scanners, animal scanners, that are much stronger. But that's what you always get with journalists, that they have the headlines that speak and the details come later. But indeed, this will be the world's strongest MRI scanner for human use, especially for whole body human use. And these are some of the news items that you saw. This is an artist impression of the MRI scanner. This was hilarious when we saw this for the first time, because this is totally not private. It's totally not private. So if a patient actually gets in here, you can see all these windows, and it's beautiful. I mean, with wood and windows and wood again, but it's totally unrealistic. It would be nice if it looked like that. On the other hand, the size of the scanner actually will be like this or similar to this, but I'll come back to that later. So my talk is going to be about 3 topics. First of all, why do we want 14 Tesla? Why do we want to move to that field strength? Then I'm going to explain a little bit about the fact that 14T is not 2 times 7T. It's different, and it's different in many ways. And finally, I will conclude with a status update of our projects and the collaborative efforts that we've had so far. So why do we want a 14 Tesla MRI scanner? And actually, it's because we want to understand the human body, because we believe that if we want to really understand or know why people get ill, why there are diseases, how they evolve, if we want to evolve medical science, we need to understand the entire human body. For instance, we need to know, why do we act or react like we do? Why does one person react on a certain stimulus in a different way than someone else does? And how does that work? How does it even work that we react? What happens then? And also, how do we get ill? Why does someone who has smoked for his entire life never gets lung cancer? And my neighbor, who is 40 years old, has never smoked one cigarette, gets lung cancer. So how does one person get ill and the other one does not? How does it work anyway? And if we get ill, how can we be cured? So what kind of treatments do we have actually? Do we have more treatments than we currently know? And why does one treatment work with a certain person and another and the same treatment doesn't work in another person? And maybe can we even prevent illnesses in the first place? That would be like the holy grail, of course, of medicine. In order to understand the body, the human body, we need to study the human body. And there are several, well, not recent advances, but several advances in studying the human body that are very important. So one is, and this is the DNA strand, one is genome imaging or genome, I totally blank out with the word, whole genome sequencing. So whole genome sequencing has opened up so many new opportunities to understand diseases, to see how genes affect diseases, how they initiate diseases, how they continue progress diseases. So whole genome sequencing and finding out the genetic underlying factors of diseases has been very important and a major achievement in medical science. On the other hand, we have histopathology, which has been around for many centuries or for many decades already, and where we can see the body on immune on a cellular level, where we can see the different kind of cells, how they interact with each other, or at least how they seem to be within a tumor, within other tissues, how it differs between patients and between tissues. And this has been the mainstay and the gold standard for many diseases. But what we miss is actually what the human body is. The human body is not a static picture. It is a machine, like this image. It is a machine where every molecule, everything interacts with each other, which is constantly in motion. There are constant new things. There are metabolic pathways that lead to certain things. There are genetic, of course, there are genetics, but they cause something dynamic, not something static. And this is something that we really have to unravel in order to really understand the human body. Well, you can say it's a nice vision, but it's not really the answer to the question that I just asked myself. Why do we want to go to the 14 Tesla? And that is because the 14T would be a very super strong magnet. And that means a much higher sensitivity. And if you compare it, for instance, to 7 Tesla, the 14 Tesla MRI scanner will have 3 times the sensitivity to the 7T. So it's not twice, it's 3 times the sensitivity. And there have been previous opinions about going to a very high field strength in the past. So I actually Googled a couple of them, and I found out, to my surprise, to be honest, that in 2015 already, Kao and al. Was discussing the image homogeneity and the signal to noise ratio and specific absorption rate for human brain imaging at several field strengths, including 14 Tesla field strength. And they stated that imaging of the human brain at up to 14T could be performed safely with advantages in SNR. So that's a good thing. Another paper from Budinger et al. In 2018 actually discussed whether you can do MRI and MRS of the human brain at a magnetic field strength of 14 Tesla to 20 Tesla even, and was discussing the technical feasibility, the safety, neuroscience horizons, and they had several areas of brain interest or neuroscience of interest, for instance, DWI, Bold MRI, QSM, CES, DCE MRI, so several areas where an ultra-high field strength could be advantageous. What does 14 Tesla have as potential? You can have a ultra-high spatial resolution, a ultra-high spectral resolution, and a ultra-high temporal resolution. Of course, just like as probably many of you know, same as for 7T and for 3T, you cannot have an ultra-high spatial resolution and spectral resolution and temporal resolution. So if you have an ultra-high spatial resolution, you won't be able to get the same high temporal resolution as well. But that's okay, because you can also have a great temporal resolution with an ultra-high spatial resolution, something that you cannot have at lower field strengths. For instance, you can scan with very high detail within the same acquisition time as on lower field strength. So what can you do with that? I think you can unravel very tiny structural details with an ultra-high spatial resolution. And you can also discover mechanisms of brain function at the circuit level, for instance, neuron cells. With an ultra-high spectral and ultra-high temporal resolution, you can visualize this dynamic disease process that I was just talking about. So not a static image, but a dynamic process. We can identify key molecular players and perhaps hopefully find new treatment targets. The main thing is we can do all of this in vivo. In this case, I'm talking about the human brain. You can sort of translate this to the entire body, but I'm a neuroradiologist, so I like the brain. I don't understand the rest of the organs, but I'm just joking. But you can all see this in vivo in the human brain in a very, very high detail. So these are some examples of details that you can see and that might be interesting, and actually might prove to be very interesting for treatment purposes. So for instance, localizing brainstem pathology, where this is the locus coeruleus in Parkinson's disease that you can see at 3T, at 7T, and this is a staining where you can see it, where you can see the minute detail. And what we hope with 14T is that we can see this, almost this, instead of this sort of, well, whitish blob that we see at 7T. And also this is ex vivo, not in vivo. So we hope to see this in vivo as well, because this can tell us something about brainstem pathology in Parkinson's disease, which is a debilitating disease that we don't really have a solution for yet. And this is an example of what the cortex looks like of the brain. So the cortex has 6 layers, 6 cellular layers, and they're altogether, they're 3 millimeters in thickness. What if we can actually see the anatomical and functional delineation of all of these 6 layers? Then we have a much better understanding. We increase our knowledge on connectivity between the different layers and cognition and how that relates to each other. And that actually comes back to our vision that we want to understand what the brain does, how the brain functions. But I think, why don't you react in the same way to a stimulus as I do? We hope that we can find that out when we look at those different layers that are so essential to connectivity and cognition. These are 2 other examples. This is all mainly high spatial resolution, but also a high contrast to noise ratio, which probably everyone who has worked with 7T knows that you don't only have a higher spatial resolution, you also have a higher spectral resolution, higher contrast to noise ratio. So you can see things more clearly because they're better delineated. All the different substructures of the tissue are better delineated. And you can also see that the 7T, we probably all know these examples where you look at cortical layers and the MS lesions within those layers that you cannot see at 3T, but you can see them at 7T, and you can compare it with histology. So we're sort of moving towards histology here when we do the 14T. And it's funny that one of our collaborators, or actually the PI of our dynamic consortium, David Norris, he mentioned, he named the 14T a mesoscope. And I actually looked up what a mesoscope was. I didn't know that in the beginning. And a mesoscope is actually, it's used for pathology. So a mesoscope is something that where you can clearly see a lot of details, but it's not as highly detailed as histology. A mesoscope is more, you can sort of see an overview. And what we do with the 14T is not making a microscope that can image the human cells. We're not going to see individual neurons, or at least I don't think we're going to see that unless it's ex vivo with a very long scan time. So that's not really realistic. But what we can do is at least zoom in more, so that we have a complete overview, but still have the detail that we need. But this is an example of white matter hyperintensity. This is also at ultra high field MRI, and you can see this at 7T and then at 11.7T. You can see there's much more detail on the T2 and T1 weighted images. And these actually look, especially the T1 weighted image, it really looks almost like it's a biopsy, compared to the 7T even. These were all static images. This was all structural imaging, but also functional imaging, the different layers of the cortex. But I also want to point you to the fact that, like I said, the body is a dynamic machine. And normally we were talking about the whole genome sequencing and about histopathology, but for these things, we're restricted to tissue. So it's oftentimes postmortem tissue or biopsy tissue as a primary source to understand the pathophysiology of disease and the progression of disease. And that's oftentimes is biased towards an end stage. So for instance, in Parkinson's disease, many of the symptoms they occur relatively late in the disease. So first there's something wrong in the brain, and then after a while, patients get complaints. But what we really want to see, we want to see the beginning, not the end, because we already know the end is symptoms, and probably you never can do something about it, or at least it's difficult. So we want to move back to the beginning, to how does it evolve? How does it actually occur in the first place? Also, this is postmortem tissue, and tissue in general is a static point in time, and they are small samples. For instance, even in a tumor, if you resect a tumor, then generally you get several different samples, and they're sampled by the pathologist. He or she is going to look at it under the microscope and see the different cells, and they can do a whole genome sequencing as well. However, this is just a part of the tumor, and generally tumors are overall very heterogeneous. So maybe in the other area of the tumor, there's a completely different profile, maybe not completely different, but also slightly different. And that different profile might actually change the way you treat someone. And if you don't have an overview of that entire tumor, but just a little snippet, then perhaps you're not really treating the tumor in the correct way. An alternative, of course, are cell cultures and animal models, because in that way you can always do imaging, because you can do imaging at 20 Tesla scanner, maybe even a higher Tesla scanner. But it has some advantages, because you can do in vitro or in vivo assessment of molecular dynamics. For instance, you can do a 19F MRI to track macrophages in animals, and you need a very small amount of the 19F probes, but you also need a very high spatial resolution. You need a very high sensitivity, and these mice need to be in the scanner for a prolonged period of time, so you cannot currently do that in humans. So you have several advantages, but on the downside is that they do not overlap enough with the in vivo human condition. And you can see that, for instance, in treatments, there are several treatments for animals for certain diseases where they make a perfect model that should simulate the disease in the human. However, in mice or in rats, the treatment works, and in humans it doesn't. Why not? Because you make a perfect model, or at least you think you make a perfect model. Apparently, you cannot really model the in vivo human dynamic condition in the same way in a non-human. That makes it difficult to actually visualize this dynamic process. So these are a couple of examples of what I mean with the dynamic body machine theory. And these are all brain examples. So apologies for the people that do not do brain imaging or are interested in more than the brain. I totally understand, by the way, or no, I don't really understand, but because the brain is great. But I'm just giving you a couple of examples about the brain. Please, I would like to remind you that this scanner is not going to be only for the brain, even though MRI and the brain are of course a perfect marriage. So one of the things that I would like to show is the cleansing the brain. So this whole lymphatic system theory, and I think that many of you have already heard about that the brain is not a privileged, not so really a privileged site for that is devoid of immune cells and immune reactivity, but it has a lymphatic system, it has a sort of a lymphatic system, it has a waste clearance system, and we're only starting to understand this whole dynamic process. And this whole cleansing the brains with the vessels, the CSF, waste clearance, that is so important. We know very little of it, but we do know that it apparently is very important in several diseases and maybe even almost all neurological diseases. For instance, Alzheimer's disease, cerebral amyloid angiopathy. And these are some images of, for instance, this cleansing and this CSF circulation system and how this actually works in practice. And there's been a relatively recent article that the meningeal layers and this subarachnoid space actually play a very important role in this whole cleansing the brain, but also in the immune system in the brain. All extremely, in my opinion, extremely interesting topics, but also interesting not from a neuroscience perspective, but from a perspective of understanding neurological diseases, understanding why things evolve the way they evolve, and perhaps and hopefully even pointing out certain areas or certain mechanisms or pathways that we can target with dedicated treatments so that we may even be able to prevent symptoms from occurring or potentially even reverse the disease. This is another example. I personally like this example because that's mainly because I'm setting up a research line in neuroinflammation, so I'm very enthusiastic about this. But it has similar overlapping things with the whole waste clearance system in the brain, and that's the immune defense system. So the immune system in a healthy brain and disease. And these are some of the key players or some of the cells that are important when we talk about neuroinflammation. And the way neuroinflammation is actually linked with systemic inflammation, so that perhaps systemic inflammatory diseases do have an effect on the brain. But how does that work in practice? Can we visualize that? And these are some of the MRI sequences that you can use to visualize certain areas of this inflammation. So, for instance, this is the blood-brain barrier. So these are the endothelial cells, and here you have water entry into the brain parenchyma. And this water entry or the permeability of the blood-brain barrier, you can visualize with several different techniques, for instance, flow studies, but also IVIM, so the intravoxel incoherent motion, DTI or DKI, diffusion tensor imaging or diffusion kurtosis imaging, DWI, diffusion weighted imaging. Then the macrophages and the microglia that are the scavengers of the brain immune system. These can be imaged with USPOs, but also with the 19 fMRI. And like I said, the 19 fMRI is generally something that we use in animals. And with the 14T, we may be able to use it in the human body in vivo and actually look at the macrophages. And of course, this seems to be on a cellular level, but we're not going to look at one macrophage. Of course, we're going to look at a bunch of macrophages that are dispersed and that can move throughout the body. And an example that I would like to give you is that there's been a relatively recent study where they injected I think USPOs, not 19 fMRI, but USPOs, so the ultra small paramagnetic iron oxide particles. And you could see that from the ventricles, they moved first towards the nasal cavity and then towards the esophagus. So essentially, you're actually swallowing your own waste. And I'm very curious to see what if you actually swallow your macrophages as well. It sounds really gross when I say it like this, but it was very interesting. And then the iron release. So because you have a higher field strength, you're more sensitive to susceptibility effects. Then the neurovascular units. And that's we're talking about the immune system, about the waste clearance, but also the vessels. The vessels and the vessels are dynamic. The flow in essence is dynamic. So the neurovascular units, the vessels with the microstructure around it, the minuscule vessels and their environment, they turn out to be very important for diseases like small vessel disease, neurodegeneration, leukodystrophies. And we hope that we can see this on a detailed overview. We can have a detailed overview of these small vessels and how they interact with their microenvironments. So, and this is a slide where that I put in because I heard there are many skeptics about this whole project and that say, well, and especially clinicians like me that say, yeah, well, will never be able to use it in the clinic. And what do you want to see anyway? And these are two examples of work that come to mind when people say things like that. And what we cannot see, we won't know. So maybe we'll see much more. Maybe we'll see things in diseases that we have never seen before because we have never done something in vivo with such a high sensitivity and looked at it from this area, from this perspective, as a dynamic machine instead of the static tissues. And this is an example of something that also, well, is an example of what we cannot see, we won't know. This is the person who discovered penicillin, and that was all by chance. It was just looking at his petri dish that he had unopened or that he had opened somewhere in his lab, and I think the window was open and some pathogen from the departments above him flew in through the window, and it was just the right temperature. And of course, these are like discoveries, and maybe we're not going to make those discoveries with the 14T, but this was something that it never occurred to him to look at this and to see this. It never occurred to him that this is how it works. And maybe, and I hope actually that we will find things with the 14T that we never thought were possible or never actually thought about at all. And this is a book that that I really recommend, that has all these kind of examples. It's very motivating when you do research, or when I don't know if there's Ph.D. students in the audience, but it's very motivating for your own research to look at the 12 highlights of modern medicine, or it's relatively old, but relative highlights of modern medicine. It makes you want to look at new things in a new way. Anyway, I'm probably running out of time otherwise. 14T is not exactly the same as 2 times 7T. Because normally in MRI scanners, we use niobium titanium. It's almost exclusively used. It's a low temperature superconductor. That means that it needs extensive cooling with liquid helium. This is the inside of an MRI scanner. You can see the liquid helium. This is another one. I don't even know where it is from, but I don't even know the vendor. But anyway, this is another one where you can see that the helium, the liquid helium pockets are a little bit smaller, but essentially you need extensive cooling. And it has a critical field of up to 12 Tesla, which means that getting a field strength of 12 Tesla is relatively difficult without problems. So then we want to do 14T, which means we cannot use the low temperature superconductors. So what we're now doing is we are switching to a high temperature superconducting magnet. And I put it between brackets because a high temperature, it doesn't mean it's like 15 degrees Celsius, that's normal. So it still needs to be cooled, but it's cooled within a vacuum. And we use, instead of niobium titanium, we use a chemical compound of rare earth barium copper oxide, which is also called REBCO. And this has a critical field of 28 Tesla or even higher, because they're already making MRI scanners with 40 Tesla, at least that's the plan. So there are several advantages of a high temperature superconductor. So for one, it has a high current carrying capacity. So you can put a lot of current through the wire. It's actually not a wire, it's a tape. But anyway, you can put a high current through it, which also means that it can be compact and it can be a light magnet design. So it doesn't have to be like this huge, enormous MRI scanner where you need like half a city to actually put it. It's not necessary. It can be almost as small as the 7T or the 3T. And then it has a stray field volume that is similar to unshielded 7T systems, which of course is very important when you think about the whole construction of your machine room and of the area around it. Then, like I said, it's conductive cooling, so you don't need liquid helium. You don't need a quench pipe. It's safer because normally you have a cryostat, which is a high pressure. Normally you have a high pressure vessel, and this is the cryostat here is not a high pressure vessel, while the helium, of course, is. And when it quenches, a high percentage of the tape that you use, so it's not wire, it's more of a tape. I haven't never seen it, so don't ask me afterwards what it looks like. But it has superconducting tape, and when it quenches, it's not that the whole tape is broken. There's probably areas that are broken and that can be repaired. So a high percentage is actually reusable. There's two drawbacks. It has a higher cost, so it's just expensive, and it is driven instead of persistent mode. So driven means that it's permanently connected to a power supply. So you will continuously need a power supply. And this is the example of the 14T and the bore size. So you can see with the increasing Tesla, so this is 79.4 two times and then 11.7, is that you can see that the bore is becoming a little bit smaller. And this is actually a wider bore that's more similar to the 7T system. And it even has the outside of the bore is even thinner than for the 7T. So what else do we need to make? Everything. There's nothing for 14T, at least not for a human body scanner. So we need to make everything. That means RF coils, transmit, receive coils, whole body gradient system, a console. We need stable magnetic field to be 0 feet. One field needs to be stable. We need extensive safety tests, and I can go on and go on and go on. It's actually making an entirely new machine, which is what we do. A brief status update and some discussion on the collaborations that we've had. So this is to make it more of a picture in your mind what it's going to look like. This is where it's going to be, and it is between in Nijmegen in the Netherlands. It is between the Radboud UMC, is on the left side, and the Donners Center for Cognitive Neuroscience. And it's the Trigon building, it's called, and that's on the other side of the street. So actually, this is the street that you see here, and this is the building, and on this side there's the Radboud UMC, which makes it on one hand convenient because there's a hospital nearby. On the other hand, less convenient because there's no direct connection with the hospital. But anyway, this is the best that we can get. We're actually using reusing a building that is not being used right now, and they're rebuilding it, or they are changing the interior and changing things so that the scanner can be there. And these are some artist impressions of what it would look like. And like I say, this is not possible because it's not it's patient protection and everything, privacy. It's just useless. But it would be nice if it looked like this with an entire room without any electronics. It would be great. It's not going to happen, but at least this is sort of what we're going to make and going to use. The magnet has been ordered, so it's now a year past after we got the grant, the $19 million euros grant. The magnet has been ordered. The design of the magnet is ready. The contracts have been signed. Apparently, that's a big issue thing. Apparently, I'm not high enough in the hierarchy to understand that this is a big issue, but it is. The contracts with the other consortium partners have also been signed, so everything is sort of ready. The console is limitless regarding the transmit and receive possibilities. And the first milestone is going to be in October 2024, so this October, because then 15% of the magnets will be built. And we can actually see whether the magnet design is feasible, yes or no. So what I've understood, and I hope that if you ask me questions and don't know, I know where I can send you to. But what I understand is that what we're going to do is put a current on the 15% that has been built already and put a very, very high current in it so that it sort of stimulates what kind of current or what kind of magnetic field would actually occur when you have the entire magnet, and if that's stable, yes or no, because that tells you something about the feasibility of the magnet design. And that October 2024, when we do that, it's going to be a go, no go of this magnet design. Either it works or it doesn't, and then we have to come up with a solution. Homogeneity tests for B0 and everything, they're not yet possible. You really need the entire scanner for that. However, we have not been idle, so you can of course simulate a lot on 14T, and you can simulate even a lot on 20T probably, but you can also simulate a lot on 14T. And these are some of the examples that have recently, one example that has recently been published about the transmit head arrays at 14T. So they found out through simulations that homogeneous excitation at 14T is possible, but that you need to think about what kind of coil element and the number of transmit channels you can use. And you have to think about the star. And what it turns out is that a uniform flip angle, which you can achieve using certain pulses, if you can achieve a uniform flip angle with these coils and with this scanner, then the head SAR and not the peak SAR becomes the limiting factor. And that's actually similar to the 70, because that's one of the safety things is, will we be able to use the scanner within the SAR restrictions? And the peak SAR is very important when you go to a high field strength. So the fact that the head SAR is actually more important is a good thing for us. Then this is the role of the consortium members, and this is actually the role of people that are co-PI and that have collaborated on the grant proposal that we now got. And there are so many people that I sent this to my colleagues, to all these other people, actually to all the people in the pictures, and they said, yeah, but you're missing this person, you're missing that person. So I just removed everyone because I'm always going to miss people, but I just want to show you that it is a collaborative effort with the Amsterdam UMC, the Radboud UMC, and all in the Netherlands, the Radboud University, the Donners Institute, all in Nijmegen, Maastricht UMC, UMC Utrecht, the LUMC, this is all in the Netherlands, and then MGH in the USA. We have several collaborators there as well. And I believe that collaboration is key in this respect. So there are 5 systems worldwide that have a field strength above 70. So we have a 9.4 Tesla in Maastricht and in Tübingen, a 10.5 T in Minnesota, and then an 11.7 T in Paris and in Bethesda at NIH. And collaborating with these ultra-high field platforms and these ultra-high field groups is essential, because they are starting to understand the safety issues on this ultra high field strength. And this is an example in 2021. I cannot pronounce his name, I'm sorry, but I think Schak discussed the long-term behavioral effects that you can see in mice that are chronically exposed to a static ultra-high magnetic field. And they show that these mice are they're not really going completely crazy, but their behavior is it's different from normal mice. Let me put it that way. However, there's a recent publication in 2023 that actually stated that when you strap the mice down, you anesthetize them, and you put them in the scanner like you do normal people, like just lying there, just prone, not moving, which they actually were moving in the study in 2021, but they're not moving, they're lying still, then those effects are not visible at all. And even if these are just two small studies and are not at 14 Tesla, there's still an indication of the safety effects and the safety issues at ultra high field. We also collaborate, for instance, with the Max Planck Institute, the Technical University in Eindhoven, and we're part of Eurobioimaging, which means that we have collaborations throughout Europe. And because this is a presentation for Johns Hopkins, Kennedy Kruger, and probably in the audience there will be mainly people from the US or from Canada, I would really like to ask you or to enthusiast you to start collaborating with us, because we need more collaborations. If there is, if you know of any a joint effort from North America, similar to the Eurobioimaging collaboration, where we can join and where we can join forces, then please let me know. And this is also something that I would like to say to everyone, join this collaboration. So think about ideas that you have that cannot currently be studied. And I was mainly talking about the brain, or actually only talking about the brain, but you can do the same thing for the rest of the body. It's all a dynamic process. The different tumors in different areas also, they're dynamic, they're not static. Think outside the box. So it's an exciting opportunity to join this ultra high field community, and I can imagine that you would like to wait until October 2024 until we have to go no go. But still, I mean, we don't really have a scanner yet, and that's true. But if we have a good idea, then it needs time to mature. And a good thing to know is that we have 90 million euros, which is an amazing grant, but it's only for the scanner. It's just the cost for building the scanner. After that, we don't have any money left. So we need grant writing. We need new ideas, new opportunities, and I would wholeheartedly like to invite you to join us on this very exciting endeavor. And with that, I'd like to thank you for your attention, and feel free to ask any questions that you have.
Can I have a video? Okay. Thank you so much, Anja. This is really exciting. Can you hear me okay?
Yeah.
Yeah. Okay. So we'll do some question and answers. Okay. Please put your if you have a questions, put your question in the Q&A box. I read the questions, and if the Q&A doesn't work, you can use the chat box as well, but we prefer Q&A if we can use that. Okay, so the first question is, when moving towards a mesoscopic resolution, would the typical microscopic contrast T1, T2 diffusion still work?
Yeah, I think it will actually. I think it will work, although, to be honest, I don't have the enough physics expertise to actually state with certainty if, of course, it's going to work. I think so, but what I noticed, for instance, is that I find it very disappointing that currently I'm working on the 7T system, on a 7T system, a clinical 7T system, and we cannot even do a flare sequence. So, and that really struck me as strange, because I thought that we could do all the kinds of sequences. It was just a different field strength, but people have told me since then that that's not really always the case. So I think it will still work. Bulls will almost definitely work. We have a whole neuroscience team that works on that. So they totally believe that Bull will work. But T1 and T2, to be honest, I don't really know. I think so. At 11.7, it also works. So why not at 14T?
Yeah, great. So second question is, how do you build a fraction of a magnet?
Yeah, I think that again, this is a very technical question. I actually talked to David Norris about this a couple of last Friday, and I
Mm,
think that actually what they're doing is that they make a fraction of the magnet, as in they make 15% of the coil essentially, and then they
mm,
put a lot of current through that 15% of the coil, and thereby they put in much more currents than they would normally do through that 15% to make sure that it sort of stimulates the
mm.
entire magnet, the entire coil. Does that sort of answer the question?
Yeah,
Hopefully.
yeah, that's great. Next question. So initially designed, you use the
Visco.
BISSCO, right? Yeah, yeah.
Yeah,
Are you
visco wire.
switching to yeah?
The
You
Repco
can say
wire, as you
yeah,
mentioned, yes, we
rapid
are
wire.
because
That
I
you
actually,
mentioned.
I
Mm,
actually put visco wire in it, and I sent it to my colleagues from the Dynamic Consortium,
mm,
and I think David immediately emailed back and said, no, 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 it's Repco wire now, it's not visco wire, and like I say, well, he explained to me the difference between the different wires, and apparently Repco wire is the second generation wire, and initially it was not as good because I think it was less stable, but now there's a company that, or now it has sort of evolved over the years, so that it's probably better than Bisco, and it's probably even cheaper than Bisco as well.
mm, mm. Yeah, this is for the magnet, right?
Yeah.
So I actually have a sort of a similar related question. So I've been sort of reading lots of news in recent years about the developments in so-called high temperature or room temperature high superconductivity, right? Do you think you will be able to sort of use some of the recent technology, or how would that affect you as you develop the magnet in the next few years?
Yeah, well, like you say, I'm not really the right person to ask.
Okay.
However, yeah, I've read those things as well because apparently they come on my Google timeline, which I don't understand at all. Apparently, I once Googled something about magnetism or something, but I did see the recent, the recent advancements in magnet design, and
Mm,
I think I sent a couple of those articles. I sent links to our group, and I
yeah.
got back, yeah, isn't it exciting? But we really haven't gotten any response about that. So I actually have the same question as you do, and I haven't gotten a response about that yet.
Right, right, right. It's such pioneering work. I guess most people,
Yeah.
that everybody exploring. So next question, you explain more about the anatomical and structural scans. Can you explain a little on the potential for improvement in the temporal resolution for functional scans?
Well, for instance, for the temporal resolution, generally, if we think about temporal resolution, we generally think about, for instance, the heart, so cardiac MRI, because there the temporal resolution is very important to see movement. On the other hand, I can imagine that the temporal resolution could also be interesting when you want to follow up on dynamic processes. For instance, um, if you inject USPOs, then you would like to have a very high temporal resolution to see where these USPOs go to. And not only on a vascular level, because USPOs generally stay, so the ultra small paramagnetic iron oxide particles, they initially stay in the vasculature, but afterwards the theory is that they're sort of scavenged by macrophages that then go somewhere, and you need a high temporal resolution to actually make these small snapshots, and make a sort of a video of where these USPOs go to, not only inside the vessels but also outside. So this is an example of the temporal resolution. So think about what can you image in high detail in a dynamic process. Sort of look at the highways, either the vascular highways or the other highways. Another example that I can give you is what I would really like to see, and maybe that's not possible, but I hope it is to see the dynamic, the dynamic process of water getting outside of the vessel and then getting inside, or the CSF that pulses, and you need a high temporal resolution to actually see the pulsation, for instance, of the CSF and where the CSF flows towards. So those are kind of things that I generally think about when I think about the temporal resolution and why you need a high resolution for that.
Yeah, great. Thanks. Next question. Who on the team has experience in building human high field magnets?
Good question. Good question,
Yeah.
Jeff.
Yeah.
I don't know.
Yeah.
Well, who on the team? Well, to be honest, well, the person that I always link with building magnets, which makes no sense whatsoever, I think, is Dennis, but Dennis Klop. But he's in engineering, and he mainly builds coils and not so much magnets. We did have a magnet. What did we call that? Um, a group of people that we asked for advice on building the magnets, and those are not co-PIs on the grants application, but we did have a group of people that we specifically asked for advice on how to build the scanner, and these were the ones that actually came with the high superconducting material and with all those kind of things. So there are connections, but to be honest, I don't really know who's in that committee. But we did have a committee, or actually, we probably still do have a committee that is experienced in building those machines, those human high field magnets.
Yeah, thanks. Another question. Moving to high resolution, head motion and physiological pulsation of brain become larger than size of the voxel. Sorry, I'm reading too quick. Do you have any plan for handling motion at 14 Tesla?
Not yet. No, not yet. No,
Yeah.
we just don't. We just don't have that yet. So what we currently have, so like I say, we have several simulations that we've been doing. And yeah, well, in general, it's also waiting for the first part of the magnet to be built and have it no go. Another thing, you have to wait a long time for contracts. It's all these bureaucratical administrative things that always pop up. Then we have one person finally that we've been able to attract for a vacancy for a postdoc position, or actually an assistant, or either assistant or associate professor position, who is going to work on those kind of things, but he will start in August this year. So for now, well, as far as I know, we haven't come up with plans yet, or probably people have thought about it, but we have no concrete plans yet.
Yeah, it must be very challenging when you go to small voxels.
Yeah, it is.
Yeah, yeah. So Jeff, I see your hands still up. Do you still have questions? You can put it in the QA box. So in the meantime, I have two additional questions if you don't mind. One is, I was wondering when you look at those mouse studies for safety, you're a clinician, so I guess this is a good question to ask you. How do you how relevant do you think to study these safety sort of profile in mouse brain? Because the more I do animal study and the human study, I realize the brain and also the body is very, very different, right?
Yeah.
Size and how they the process of the physiology. We do, for example, we do olfaction study in mouse and the humans that complete different. Often we find something in mouse, but we cannot reproduce in humans. Lymphatic studies as well, and all those structures are so different. So what's your opinion, for those mouse studies? Should we move to more human studies, or how relevant are those results as you show to different studies?
Yeah, good question. I am not in favor of animal studies at all, because I think they do not really reflect, like you say, they do not really reflect what the human brain does, how it reacts to certain things. So I agree with you. I'm very hopeful for the next step that, for instance, they're going to do either in Paris or at NIH at the 11.7, where they just put healthy volunteers in it. And
Yeah.
I think that's our not our holy grail, but that's something that I'm very interested in, because to be honest, I don't think that the effects of the 11.7 Tesla magnetic fields will be that much different from the 14 Tesla, but maybe I'm naive, but I don't think that's going to be that much of a difference. So if we have good results at the 11.7, then I feel a little bit more safe about the 14 test lab. And because that's on humans. On the other hand, if I was in Paris or at NIH, I would really be quite, well, I don't know, not afraid, but I would be sort of excited and scared at the same time when the first volunteer got in. Yeah, it's true. Yeah. I mean, I don't know what can actually go wrong. I don't think that much can go wrong, but on the same time, we don't really know that yet, right? I mean, we do know it in mice, so there's not that much extreme that's probably going to happen, but still.
Yeah.
I'm glad that we have an 11.7 and that people are scanned on that.
Yeah, great. Thanks. So one last question. I think you mentioned in the end, you mentioned that you're looking for potential collaborations, grant idea, those things. I guess those are limited to European people in Europe. And if not, can you give us some examples just to inspire people who might be interested here?
Well, I mean, I don't have that many examples on direct collaborations within this proposal. Let me put it that way. So the only person, there was only one group that actually worked with us on this proposal initially, and that was from MGH. So that is American, and I don't think that we have other active collaborations right now, but that's mainly due to the fact that we have a grant from the Netherlands. We needed this was an infrastructure grant where we needed to show that we could make this infrastructure and build this machine. So it was a very specific reason of doing this, of having a European aspect in it as well. And that's why I sort of closed my presentation with this, that I think that it is not restricted to Europe. It's actually, this is a scanner that can be used by everyone all over the world. And that's why I'm reaching out to you and saying, please email me. Let me know if you have an idea. Let me know if your institution wants to work with us and work on this project, because this is a system. There's not going to be that many other 14 Tesla systems. And if we can collaborate together, then we can make this machine work and do great science with this, and hopefully find new treatment opportunities and things like that. And I think that especially when we do it with all of us, and there are many ultra high field platforms within the US, within North America, but also in outside of America and outside of Europe. I think that if we join forces, then we can actually make this work much better. And that's also why I asked, is there something like that Euro bioimaging consortium thing that is also active, but then a similar thing, but then a North American one, for instance? We discussed this entire project in the ultra high field group of the high field imaging group of the ISMRM. So we try to involve as many people as possible. And this is another way of doing that.
Yeah, this is really exciting. Great. Well, thank you so much. I guess initially I thought it might be shorter, but it turned out to be such a great presentation and lots of great discussion as well. Thank you so much, Anja, for this great presentation and all these information. And thank you everybody for attending this month. We'll see you next month. Thank you.
Thanks.